next topic, that is interaction between the genes and the environment, right? Okay. So for that, we will take an example of a operon over here, right? I hope you have already learned, right, in school.
Yeah. We've
Okay.
done that.
So can you
operon before.
explain me what an operon is?
Yeah. So basically, it's like a bunch of genes, right? That are like from the same kind of like they first of all they're close together and they all have like the same promoter sequence. I remember that.
Try to understand, Akash, over here that
Mm-hmm.
let's say, and one more thing, you must have an idea about that sequence also, expression of gene also. We have learned that how a gene express itself by undergoing transcription and
Translation.
translation, right? Okay, cool. So what happens is, let's say this is your DNA, okay? And on that
Mm-hmm.
DNA, what happens? Let's say we have a gene, right? This is our gene. Let's say gene A, okay, clear. Now this is the gene which
Mm-hmm.
we want to express, or the cell or the system wants the gene A to express itself, right? Okay. So that part of DNA which has to perform transcription, the part of DNA which has to perform transcription is called transcription unit. What it is called?
Transcription unit.
Transcription unit. Okay. And this transcription unit is actually uh, marked by certain boundaries, right? That from which point the transcription will start and the point where transcription will stop. So the point where transcription starts is known as promoter, and the point where transcription stops is known as terminator. Okay, what is P here?
P is promoter.
P is promoter and T is
Terminator. Like the movie.
terminator.
So P is
Okay.
for promoter.
Yes, P is the point
Okay.
from where transcription will start, and T is the point where transcription will stop. Now,
Okay.
in between P and T, what you have the DNA sequence that will undergo transcription and it will form your mRNA. What it will form?
mRNA.
mRNA. Right? Any doubt, Akash?
Nope. No, sir.
Now, along with these sequences, there are some other DNA sequences or genes that regulates the functioning of this gene, means whether it will be on or off, right? Okay, that is actually regulated by some other set of genes. Okay, so let's say we have a I gene over here, that I gene, along with that we have a O gene also. Okay, clear? Now, along with the regulatory genes, along with what? Regulatory gene, that entire structure is known as operon.
Yeah. Oh, like
Okay,
that. Okay.
clear? So what actually operon is? Operon is your transcription unit, which involves your promoter, a structural gene, and your terminator. Okay, this is your transcription unit. Along with what? Along with your regulatory genes. Which genes?
Regulatory genes.
Like, for example, gene I here and gene O here.
Oh.
Understood?
Yep.
Okay, so this gene I, right? Okay, so this gene I, what it does here is it produces certain, again, it will also undergo transcription and translation. So after translation, this synthesize some proteins, right? Now these proteins either activate the transcription unit or you can say a structural gene, or they can inhibit the structural gene also, right? Means your gene can be switched on or switch off. Okay, using these proteins. Clear?
Yep.
Yes.
Okay, fine. So here we first time remember some names are also given, right? So Jacob and Monad. These Monad, it's not Monod, it's Monad. So Jacob and Monad, these are the two people or the scientists who were the first one to came up with the concept of operon, right? So we need to discuss about lac operon. Now, what does lac means here? So basically, what lac over here means is lac means lactose. What does it mean?
Lactose.
Lactose, right? So lac operon is your gene that is responsible for lactose metabolism. This lac operon is responsible for what?
Lactose
Lactose
metabolism.
metabolism, right? So whenever the organism, the bacteria, what was the name of bacteria which has this operon in the
E.
previous
coli.
slide? E. coli, right? Okay. So generally for E. coli, let's say this is E. coli, a bacteria only, right? Okay. What is their preferred food?
Bacteria preferred food like this. I mean, I don't know.
For them,
Sugar,
it is
lack of sugar
glucose.
should be good.
Right. Okay. So simple sugars like glucose, right? This is their preferred
Mm-hmm.
food. Okay. So if they have glucose or any other sugar in the medium, they will always prefer glucose because glucose is the simplest form of sugar. They can break it down and they can get energy out of it, right? But imagine in case
Yep.
if glucose is not there in the medium, right? And what we have is
Mm-hmm.
lactose. If in the medium, what is present?
Lactose.
Lactose is present. Now, when this lactose is taken by the bacteria, there must be some enzymes present in the bacteria to digest lactose, right? Okay, because lactose is not a simple sugar. Lactose is what? Lactose is a diamine. So we have to
Disaccharide.
break it into, yes, disaccharide. So we have to break it into its constituent in order to get energy. So we need some enzymes to digest the lactose, and those enzymes will be synthesized using which operon? Lac operon.
Lactoferrin.
Okay, clear? So here we have two situations, one in the absence of lactose and other in the presence of lactose, right?
Mm.
What is the first situation? In absence of lactose means when lactose is not present. So when
Mm.
lactose is not present, do we need lac operon to work?
Not really, no.
No, right? Okay. So lac operon is not required. So we need to keep this lac operon switch off, right? Any doubt?
Mm. No,
Okay,
sir.
now let's see a detailed structure of your lac-operon. So lac-operon has I gene, which is your inhibitor gene. It has a promoter sequence. It has an operator sequence. Their structural gene has three different type of genes. Okay, and then you have terminator. Okay, so this is your the structure of your lac-operon. What are the genes we have? I gene, P gene,
Okay.
O, Z, Y, A, and T. Okay,
Mm.
here P, you know, right? What is P? P is promoter,
Yeah.
right? T is
Agreed. P is promoter, T is terminator, and E
I
is the
is
I is one of the regulatory genes.
regulatory gene. In this case, it is a inhibitory
Inhibitory.
gene. Okay. In the absence of
Yeah.
lactose, what we want to do is we want to keep this operon switch off, right? So what will happen? Your I gene will undergo transcription to form a regulatory mRNA. To form what?
Mm.
Sorry, regulatory
Mm.
mRNA or repressor mRNA also you can say. Okay. Then
Mm.
it will form a repressor protein. What it will form?
Repressor protein.
Repressor protein by translation. So let's say this is a protein. Now what this protein does is it goes to the operator and binds with operator. Binds where? With the
Binds the
operator.
operator.
Right. Now because of this binding of protein with the operator, the main enzyme for transcription. What is the main enzyme for transcription?
Main enzyme for
RNA
transcription is RNA polymerase.
polymerase, right? So RNA polymerase couldn't
Yeah.
bind to promoter. Okay,
Yeah.
clear? Now RNA polymerase cannot bind to promoter, so what will not happen?
That promoter is not going to get
Transcription
transcribed.
cannot take place, right? What we learned that promoter is the site from where transcription starts. And for that, RNA polymerase must bind to the promoter site, right? Now, as RNA polymerase cannot bind to the promoter site, so transcription will not take place. So no transcription means the gene is switched
Mm. Off.
off,
Off.
right? Any doubt?
No.
Okay, cool. Now this is the situation which is we are talking in the presence of lactose, right? The lactose is present in the medium. So this is what?
Mhm.
This is your lactose molecule. Now what happens is the lactose which is present in the medium, right? This lactose acts as a inducer, right? So this lactose,
Mm.
what it will do, it binds with the repressor protein. It binds with what?
Repressor protein.
Repressor proteins. Lactose binds with repressor protein. And because of this binding, the protein becomes inactive. Okay, protein becomes
inactive.
right. And what protein was doing actually? What was this protein actually doing in the first case?
It's like repressing, right?
Right. So this protein, it was doing, it was binding with the operator. Right. I'll write it down here. This repressor protein binds with operator. Now, as it binds with operator, what happened? RNA polymerase couldn't bind. Okay. Now, in this case, when this protein is now inactive, what will happen? There is nothing to bind with the operator, right? So promoter site is free. So as the promoter site is free, what will bind with promoter site? RNA polymerase. Okay. So RNA polymerase binds with promoter, and what it will do? It will initiate transcription. Okay. So RNA polymerase binds with the promoter site, transcription will start. So all these genes, gene Z, Y, A, they will undergo their mRNA. And then after that, they will undergo translation to form their final enzymes. And those enzymes are required for lactose metabolism. Clear? So in this case, the gene is switch on. Understood? Any doubt?
Then now.
Okay, so let's
No, sir.
recall. In the absence of lactose, what happened was the I gene produced repressor proteins. Those proteins binds with operator, and because of that, RNA polymerase cannot bind with the promoter. Now, as RNA polymerase cannot bind with promoter, there will not be any transcription. So no transcription means no enzyme will be formed, no, basically no protein will be formed. So that gene is considered as switch off, right? Any doubt?
No, sir.
Now, in the presence of lactose, what will happen? Again, the repressor protein will be formed by your gene I, but this time the repressor protein will be joined with lactose, and that will make the protein inactive. Okay, so now RNA polymerase gets a chance to bind with promoter, and then transcription will take place as usual. All the genes Z, Y, A, they will synthesize their respective enzymes. What are those enzymes? Gene Z codes for? Any idea?
Could you read the question again, sir?
Gene Z codes for which enzyme?
Honestly, no clue.
So can you repeat the question again? Sorry.
Gene Z codes for which enzyme? Okay, gene Z codes for beta galactosidase. Which enzyme? Beta galactosidase.
I
Y
would have never
codes
guessed
for
that.
permease,
Literally.
and then trans-acetylase. Now, permease is the major enzyme which is responsible for lactose metabolism, basically the breakdown of lactose. When we break down lactose, what we get? Glucose and
Maltose,
galactose.
galactose.
Right? Okay, now studying variations in human beings, right? So basically, what are variations?
Variations are differences.
Mutations, essentially, that cause phenotypic differences.
Right. See, basically variations, as you said, the differences between us, right? Okay. Not only us like family members, between each and every individual, there are differences, right? Okay. So there are certain terms we have discussed earlier, if you can recall, right? Akash, phenotype, genotype, right? Okay. So what was phenotype? The
Physical
physical
expression of the
appearance,
particular
right?
alleles you
Physical
have.
expression, right? And that phenotype is actually dependent on what?
The genotype.
The genotype, right? Clear. But the genotype is not the only thing that decides the phenotype. Okay, there are a number of other factors which can also contribute to the physical expression, which involves your environment, right? Your exposure to different things. Okay, so this in this particular topic, this is what we are going to discuss, that how phenotype can also get varied. Here,
Yep.
okay. So genotype along with environment also plays a very important role in the expressions of the genes, right? So some studies have been done in the case of human beings also, and a very good subject material that we can select for human beings are identical twins, right? So let's say that we have an identical twin. What are identical twins?
Same genotype.
Right. Okay. Means these are two individuals with the same set of genes, same DNA, right? Clear? So in the early stage, what we do is we make them separate, right? And what we do is we place them in two different habitats, or you can say in two different environments.
Hmm.
Okay. Clear?
Yeah.
Now these two environments are different, right? Now, if DNA is the only factor, right? So in different environment also, they should not have any variations in them when they are growing up, because same DNA they have, right? So they should not have any changes or variations among themselves. Yes or no?
Yes.
Okay, but what we have seen is or observed is that when we separate the identical twins also to two different environments, what we will observe is we will observe some phenotypic changes in them based on the environmental conditions, right? Okay, so environment plays a very important role in the expression of your genes. Okay, clear? So when we talk about the influence of phenotype, right, it is not only the genetic factors, but environment also plays a very important role, right? So what are the factors that influence your phenotype? First thing is your genetic factor. Other thing is your environmental factor. Okay, clear?
Hmm. Clear, sir.
So at the end, we can say that phenotypic variation can be explained by a combination of genetic and environmental factors. Okay, clear?
Yeah.
Any doubt?
No doubt, sir.